morning and welcome to SMMF. It's Wednesday, September 26th, and it is a lovely day. Today we are going to be reporting on the cell membrane. Let's begin with the structure of the cell membrane. It has recently been discovered that the cell membrane is made up of phospholipids, which are lipids where only one of the fatty acids has been replaced by the phosphate group. The cell membrane is... Hang on. What is that? Oh my gosh, I have something coming in. Holy cow, let's go to Sarah, who is currently inside at the cell membrane.
Hello, I am here on the intracellular side of the cell membrane, whereas you happen to be on the extracellular side of the membrane. I have discovered how it is made up and works. Right here we can see that the cell membrane is made up of phospholipid bilayer. Its two layers are arranged according to their properties. Now, the phospholipid bilayer is made up of two parts, the head and the tail. Also shown here. The head is on the outside of the membrane where it faces the watery environment and is hydrophilic, while the tail is on the inside, isolated from the watery environment and is hydrophobic.
So are you saying that the head is polar and the tail is non-polar?
Yes, Maggie, that is exactly what I'm saying. Since the head being hydrophilic, meaning it loves water, it faces water, which is also polar. However, the tail being hydrophobic, meaning it's water-fearing, it is non-polar, so any water that becomes near is not mixed well with it.
Wow, that means that the phospholipid bilayer is amphipathic, which means having hydrophilic and hydrophobic parts. Did you know that there are enzymes placed throughout the membrane to enhance the rate of reactions at the membrane level?
Not only that, but as you can see right here, there are proteins that are embedded throughout the membrane. Here are the integral proteins, which are amphipathic and are embedded in the plasma membrane. And here are the transmembrane proteins, which are a type of integral protein that spans the entirety of the cell membrane, which are permanently attached like channels and carriers. This peripheral protein is not transmembrane because it does not span the entirety of the cell membrane. Do you know what these are?
I do. Protein channels allow for passive transport of substances between the inside and the outside of the cell. Passive transport means no energy is needed for the transportation to occur. If you look, there are carrier proteins. These carrier proteins bind to substances on one side of the membrane to change its shape to the other side, similar to molecular transport. It is done through active transport, which means that energy is needed for the transportation to occur. Carrier proteins use that energy to change the shape and are known as protein pumps.
Although receptors aren't always transmembrane, an example of a transmembrane one is the glycoprotein. Receptors are types of cells that span the whole membrane individually to relay information from the inside or the outside of the cell, whereas here are peripheral proteins, which could be enzymes, which are attached to the outside of the plasma membrane.
Are peripheral proteins polar?
You know it. Do you know what this is? It's a glycoprotein, which is a protein and a carb that are attached together. They are important in maintaining the structure of the cell membrane and in cells differentiating between self and non-self cells.
I think an example of glycoprotein is recognition. Recognition consists of certain proteins that help cells in differentiating between self and non-self cells, which are important in triggering an immune response.
Yeah. Whoa, what's this brown thing? I have never seen that in the cell membrane of a plant or bacteria. I'm currently in the animal cell and I see this piece of the membrane that is helping to maintain the structure. Maggie, do you know what this is?
That is cholesterol, which is a steroid only found in animal cell membranes.
I didn't know there was a component of the animal cell membrane that wasn't in plants.
Oh, God. Wow, thank you, Sarah, for venturing into the cell. Well, that's all the time we have for you today. See you next week when we'll be doing a million more labs.